Hello, everyone, and welcome to another EAStalk webinar. My name is Anna Viskovic, and I'm delighted to be your host and moderator today. Thank you all for joining us. As global food systems face increasing challenges from population growth and environmental constraints, aquaculture continues to be recognized as a key part of the solution. Today's webinar will focus on exciting and innovative initiative, the EU-funded INNOAQUA project, which has been running for 18 months and is exploring the integrated use of algae in sustainable aquaculture practices and high-value food applications. We will dive into how the project is coupling algae and fish production, particularly with species like salmon and sole, as well as the potential uses of algal extractions in various industries. We will also highlight the importance of international collaboration in developing new food products based on algae. Before we begin, just a few quick notes. The webinar will last 45 minutes with a 30 minutes presentation, followed by a 15 minutes Q&A live. This session is being recorded and will be available on the EAS YouTube channel for 1 month. After that, it will be accessible only to EAS members. If you are not yet an EAS member, I encourage you to visit our website and learn more about membership benefits, including access to past EAStalk webinars and presentations PDFs dating back to 2019. We are fortunate to have with us today 3 experts, each bringing unique insights into different aspects of microalgae research, aquaculture, and product innovation. Dr. Dorinde Kleingris, Principal Investigator Microalgae at NORS and Associate Professor at University of Bergen in Norway, with a background in bioprocess engineering from Wageningen University, Netherlands. Her research focused on microalgae strain selection, cultivation, and techno-economic assessments. She also has extensive experience in managing EU and national research projects. Rui Pereira, head of the Seaweed Division at A4F, Algae for Future, a Portuguese company at the forefront of algae biotechnology. With over 15 years of experience in the field, he has been involved in more than 20 national and international projects, and has co-founded Alga Plus, a company dedicated to sustainable seaweed production. Dr. Maria Roca Ayats, a senior product scientist at Alchemy, a Spanish startup specializing in microalgae for the food and cosmetic industries. With a strong background in chemistry applied to sustainability and green energy, she has worked in both public and private research, bringing expertise in product synthesis and characterization. Together, they will provide a comprehensive overview of INNOAQUA innovative approach and the future of algae-based food solutions. Now let's get started. I'd like to hand it over to our first speaker, Dr. Dorinde Kleingris, who will introduce the INNOAQUA project and its key developments so far. Dorinde, floor is yours.
Super. You can see my slides?
Yes, we can.
Okay, then I would like to start by quickly introducing the project, and then we'll dive into the details and the results so far. So as Anna already mentioned, INNOAQUA is very much focused on making European aquaculture, and especially land-based aquaculture, more sustainable and diversified. And we do that by using algae and integrating them with fish production for the production of novel foods. So it's very much centered around this technological backbone, where we will be looking at integrating salmon and algae production, or fish and algae production. So there's a demonstration 1 on salmon and microalgae, and then a demonstration plant 2 on sole and seaweed. And then all the products being produced, so that's both the algae and the remainders of the fish, they will be processed and turned into novel food ingredients. And then to support this technological backbone, we also focus on social innovation, making sure that we develop products that are actually accepted by the consumers and wanted by consumers. And we also use sustainability assessments to guide us in developing these processes and these products to ensure that they're really sustainable, both from an environmental and economic and a social perspective. And of course, because we're using the circular approach, we also look at the safety and regulatory assessments to ensure that we align with policies, or that we help developing policies to make sure that this will be possible in the future. And then last but not least, we also have an extensive outreaching strategy, such as today, telling everybody about the results in the project, but it's also working or looking at developing workshops, educating people, and searching this international cooperation. So as mentioned, today we'll be focusing around 3 aspects. The one that I'll start talking about now is the demonstration 1. So that's the integration of salmon in recirculating aquaculture systems together with microalgae production. Then Rui will address demonstration 2. And then Maria will follow up with the downstream processing into the novel food products. So let's go to demo 1 and the coupling of microalgae with salmon. So maybe a bit of background first. We have this demonstration in Norway, and Norway is one of the world's biggest producers of salmon and trout. So in 2022, there was 1.6 million tons of fish being produced. And then with that, they also have a very high goal set for 2050, where they foresee that they would like to produce sustainably more than 5 million tons of fish. But with the production of this fish, you don't only have the fish, but you also have a lot of sludge being produced. And not all of that is visible, because it's done, as you can see on the picture on the right, often in sea-based systems. But with all that sludge, there's also a possibility for negative environmental impacts by, for example, causing eutrophication. But it's not only that, it's also that we lose a lot of interesting components. So all that sludge in 2022, for example, that equated more than 20,000 tons of nitrogen and 13,000 tons of phosphorus. And also the carbon in that sludge, it is estimated that it could contribute to producing more than 1,000 to 3,000 even gigawatt hours of electricity. So to put that a little bit in perspective, in Norway annually in fertilizer, in agriculture, there's around 9,000 tons of phosphorus being used. So that means less phosphorus than actually is produced or is a side effect of aquaculture. And also this electricity would be enough to cover a city of Bergen, which is the 2nd largest city in Norway. So there is a lot more attention nowadays on capturing sludge. That is both in the open cages that we saw in the picture before. There's methodologies developed in capturing the sludge from those cages, or developing cages that are fully closed. But also there's an effort going towards producing more salmon, or having the production process of the salmon that starts anyway on land, but having that longer on land before the salmon go to sea, or even just fully on land. And especially work is done on doing that in recirculating aquaculture systems. So I guess many of you know, but in recirculating aquaculture systems, the idea is that the water used for the fish production is recycled, going through a set of treatments, and then depending a little bit on the operation, but 5 to 10% of the water needs to be or is released and refreshed, but all the other water, so more than 90%, is actually reused in the production of the fish. And with that 5% that we had here, there's still some nitrates lost, and the concentrations are relatively low, but at the same time, it's still very large quantities that are being produced of water. So that means that there's still quite a lot of nitrogen and phosphorus that is lost in this way. So with that, we had the idea of trying to see if we could integrate the production of microalgae that are very good in taking up nitrate from water and phosphates, and using the potential lost nutrients instead. So first we would do this, or we're doing this at pilot scale, I would say, at Marine Holmen Raslab. And then the idea is to take that process and scale it up to even larger size at Viking Aqua. Viking Aqua is a producer of fish that is planning to produce around 30 tons of fish per year, and really up to the full size salmon ready for consumption. So, as I mentioned, we're combining this process, and the nitrate amounts in the water are, and also the phosphate amounts in the water are relatively low, but there's a lot of water being produced. So one of the aspects that we wanted to look at is how can we ensure that we can take up all these nutrients? Because if we normally look at industrial microalgae production, we actually use concentrations of nitrate and phosphate, so the fertilizer for the microalgae, in higher concentrations to achieve high concentrations of algae in our reactor, and with that achieve an economically interesting process. So what we did here is that we actually took a filtration after the production. So you see here in the middle in the green, that's the algae production reactor. And from there is algae being pumped through a filtration set and up concentrated. And the up concentrated algae are being fed into the reactor. And the water that is taken away can then leave the reactor. So this way is a way of treating way more water and thus providing the algae still with a very high amount of nutrients. But not necessarily having a very dilute algae culture. So in principle, the idea was a win-win, so to say. So we tested this setup with a full production cycle of salmon. And as salmon starts in freshwater, we used a freshwater microalga chlorella in the beginning, and then we went to the salmon production process, went to brackish water. There we used pheodactylum, and then last we went to a saltwater production phase, and there we used the microalgae nanochloropsis. And what we can see here is that in the top we see the concentrations of the nutrients, so the nitrogen from the nitrate, the phosphorus from the phosphate, and then the phosphate was both in, we added a little bit because we knew it was going to be too little, because as you can see in the gray bar, that's the phosphate that was originally from the water, and then in the light blue bar, that's the phosphate that we added to make sure that the algae were not limited based on the amount of nitrogen that they were getting. And as you can see, with the fish being very small, there's less nutrients in the water, and then as the fish grow, there's also more nutrients in the water, and that also allowed us to have higher algae concentrations in the water, while using or having the 75 liter per day of RAS water flowing through. So this was nice and high algae concentrations that we're kind of used to in producing microalgae at semi-industrial scale, so to say. And then if we look at the nitrogen removal rate, so to the left top you see now the nutrient removal with in pink the nitrate and in blue the phosphate. And as you can see, in almost all cases, the nutrients were removed, well, varying from 80% to 100%. And what is causing this is this slightly imbalance between nitrogen and phosphorus. So that's something that we need to start now adjusting. So we have one partner working on sensors for measuring continuously how much nitrate is in the water, for example. So in that way, we can then hopefully dose exactly the right amount of phosphate with what the algae would be needing. So overall, we can say that the microalgae grew very well on the RAS effluent water. We reached very high microalgae biomass productivities, similar to what we would be reaching on commercial fertilizers. And we can achieve 80 to 100% of the nutrients removed, where we foresee that with better process design, we can go up to 100% all the time. So for that, we need some system improvements. The other thing that we're going to look at is also what is the impact of doing this on the biomass quality. That's both looking at bacteria, both in the algae culture, but also those that are living with the fish. And for example, heavy metals. We know that there are some metals that are in the fish feed that might leach out. So how would that impact the algae, and would they be accumulating those or not? And then, of course, we'll be doing the sustainability analysis to ensure that we also understand what are the impacts on the costs and the environmental sustainability. So with that, I would like to very much thank the team that made this all possible and that worked on all these projects. And I would like to thank all INNOAQUA partners that are part of the whole project. And with that, this is our contact details, and I'll give the word back to Anna.
Thank you very much, Dorinde, for great presentation. Now is the time to Rui Pereira to present his work. So Rui, feel free to share your slides.
Okay, good afternoon, everybody. Pleasure to be here. Can you see it already?
Yes, we can see it.
Okay. So like as Dorinda was already mentioning, I'm going to be talking to you about what we can call like a sister task from the previous one. So it's basically the same concept, but now we are applying the INTA concept to a sole production and using macroalgae or seaweed instead of the microalgae, as Dorinda was mentioning. Let me just give you also a little bit of background, because I know a lot of our audience is probably expert on aquaculture in general, and I know from experience that when we talk about aquaculture, a lot of people immediately thinks about animal production, and in particular about fish. And I would like to put you in perspective what is the weight, the importance that algae already have in terms of world aquaculture. Okay, this is the latest data from FAO. I'm not going to discuss now if there are better data or worse data than FAO, but it's official data from 2022, and you can see that in the global aquaculture, algae represents around 17% of the world production. Okay, so in 2022, that was totaling 36.4 million metric tons of fresh weight algae. On the right-hand side, you have a small chart pie that tells you what is the contribution of the European Union or Europe for this production. It's basically nothing, as you can see. So most of the production of algae in aquaculture is based in other places, but not in Europe. But this does not mean that Europe does not use the algae. Okay, actually, the area imports a lot of algae biomass. Okay, not for food and feed as it happens in any other countries. Now working exactly on that development, but Europe already imports a lot of algae biomass for other processes for extraction and using algae as phycocolloids in many other industries. So even though we don't produce it, it's not meaningful. On the contrary, it's a very important biomass, important product in Europe. Also interesting to note, oops, sorry. Also interesting to note that, is it changing to you? Sorry. Okay, now it does, okay. Interesting to note that out of thousands of algae species and seaweed in particular described, all that biomass that I mentioned before, 36.4 million metric tons, actually comes from just around 8 genus of seaweed, okay? So there's a lot to exploit. These genus are, however, most of this biomass is actually produced in aquaculture. So if you consider the groups before, the fish, the algae, the crustaceans, and so forth, the algae is actually the group where there's higher contribution from aquaculture. There's virtually almost nothing being harvested from the wild in terms of algae, and 97% of that biomass actually comes already from aquaculture. It's valued in a very significant value, as you see there, but also important that currently most of seaweed aquaculture is essentially based on production at sea. Okay, and that's probably one of the reasons why you see that that pie, the chart pie that I showed you before, in which the production in Europe is meaningful. That's because production at sea is based traditionally in these kind of technologies. Okay, so these are images from South Korea, from China, from Philippines, and Asia. It's actually dominant in terms of production of seaweed in the world, and these kind of systems will never be possible in European waters, okay? Because the geography is not there, the conditions of the sea, but also, and very important, because of regulations and lack of licenses to perform large-scale productions like these. Some of these areas, as you can see the picture on the left-hand side, it's a satellite image. So there are thousands of hectares of production areas for the seaweed production in these countries. Something like this, again, it's not possible in Europe. So if we want to develop the seaweed industry, we have to use other kinds of technologies. And the fact is that there are already other kinds of technologies for production of seaweed. As I mentioned before, most of that biomass is produced at sea, but even those productions, they sometimes require a land-based facility that works as an hatchery, let's put it this way to facilitate the terminology, as an hatchery, a maternity to produce the biomass that is then produced at large scale at sea. So land-based aquaculture is already a reality, if nothing else, to provide the source of biomass for the sea-based grow out. But it's also possible to actually produce or have a full production chain in land-based systems, either outdoors or indoors, in earthen ponds or in photobioreactors, as I mentioned there. The difference is that this will be done at smaller scales when compared to the sea-based technology. More intensive productions, so that means that a land-based system will be able to produce more biomass per square meter, for instance, or per unit area. It will obviously allow for a much higher degree of control. So land-based or sea-based production systems, when you put your material in the sea, you just have to wait for Mother Nature to do its work, and they will just grow. There's not a lot of control that you can add to the system. But if you are producing inland and a system like I'm going to show you, then you will have the capability to actually control and manipulate some of the conditions and have that higher degree of control, as I mentioned there. So land-based systems look a little bit like this. These are just a few examples of companies that exist or projects around the world. Up on the left-hand side, this company called INNOAQUA, partner of our project. They are already producing and using the uva that they're producing there as a source of raw materials for bioplastics. Still on the top, a picture from Acadian Sea Plants, a Canadian company producing land-based algae for decades now. And on the right-hand side, there are other examples from EAStalk project, the EU project. This is a facility in South Africa, actually. An indoor example of an indoor system from a Danish company. And on the right-hand side, on the bottom, it's Israeli company, Sikura, that also produces land-based systems and land-based, seaweed in land-based systems. So this is the kind of technologies that we want to apply in our demo too. So the objective of demo 2, in some ways similar to the objective of demo 1 that Doreen was also mentioning, but we add to this a different level or a new level of novelty, that is the technology development or the application of digital solutions to help in the management of these demonstration units. If you are successful in doing this, we also hope to at least start or give the first steps in applying these technologies also in our demo 1 in Norway, as Doreen was mentioning. So what are we doing? This demo 2, we are installing this land-based seaweed production at the premises of another partner of INNOAQUA, of course, C8. C8 is an aquaculture group with different facilities in the north of Portugal and north of Spain. They are now just opening and starting to work with their third on-growing facility. And we will be producing our seaweed, receiving water from their hatchery, located at this point with a circle there called Safistela in the north of Portugal. So we will basically have a flow-through with open system photobioreactors receiving water constantly from C8 hatchery, and this will be capable to deal or treat approximately 40 cubic meters per hour maximum capacity. This is the KPIs that you see there on the right. This is not actually the total capacity of the facility, of course. This is roughly 1/3 of the actual water volume that is moving constantly in the system. We have the aim of being able to remove up to 30% of nitrogen and phosphorus in the system, the total system, and based on the productivity of the most productive species that we use, we are hoping to produce around 500 to 600 kilograms of fresh weight biomass. The system will be adapted to work with different species, and the site will also include, because remember, we also have a sustainability work package in INNOAQUA, and we are worried with that sustainability aspect and LCA studies. On our site, we'll also have a pre-processing stages. So the first, even though most of this biomass will then go for extraction of protein and other compounds that you'll see in the next talk, we have also pre-processing stage in here where the biomass will already be dry and milled. so that when we transport the biomass from here to the other locations, we're not actually transporting water and make the whole system much more efficient. So what makes this more interesting or more novel, let's say, in terms of the production of seaweed, is to add this complexity of digital technologies. We have a partner that is developing on one side a different or a novel sensing model based on fiber optics, essentially, that will be able to give us real life information on key physical chemical parameters of the water. Basic things like temperature, of course, the pH, the light, and very important, the nutrient concentration. And we will use in this case nitrate as a proxy, because it's actually the form that is more present in the effluent coming from C8. And we use all these to give information to an input-output models. Of course, these models also be capable of working with sensors that are already commercial sensors off the shelf. Okay, in case the sensing model development does not reach the desired stage, we'll be able to work with off-the-shelf existing equipment. We hope the ones that we are developing will be more accessible and more efficient. And this will give on-site information to an edge processor that will give the operator information on how to better manage the system of seaweed production. So the idea is to be able to receive also information aside from the live, is to be able to automate some of the actions of the valves, for instance, receiving orders, automatic orders to open or close in case the nutrient in the tank is lowering or it's higher. But the system will also be prepared from the start based on theoretically functioning to set priorities and hierarchies in terms of which parameters will be governing the system at one point. So for instance, you as manager of the system will be able to choose between optimizing production or production of biomass or optimizing treatment of the water. So you can set this one or the other as priority. And the system will also be able to automatically change the priority. And so if at one point the level of nutrients in the water is giving the order for the valve to open or close, at a different point, the temperature may become a key factor and will actually override the information about the nutrient availability, and that becomes the priority. So this is the kind of systems that we are looking to develop. All this will be complemented with information technology, so with a cloud-based system that will connect with the on-the-ground, let's say, facilities, and will also give information based on historical and learning, and machine learning systems will give also information that will try to predict or to make suggestions on how to manage the system in the upcoming days or the coming weeks, also based, for instance, on the forecast of the weather, the expected number of hours of light in the next few days, and so on. So instead of, for instance, harvesting today or tomorrow as you were expecting, the system can actually give you a suggestion. It's going to be sunny the next few days, so with these conditions and with good light conditions, you can actually delay your harvesting a couple more days, and you'll be able to harvest an X amount more of kilograms. This is the kind of system that we are trying to develop here. If we are successful, the main goal is to actually turn these into a yes.
Sorry for interrupting. Yes.
Last slide. Last slide. So the main message is that land-based seed aquaculture, even though existing, is still a relatively low-tech system. We're trying to make these more user-friendly technology. So moving from MTA 2.0 to 5.0, it's using the digital technology, artificial intelligence, and machine learning technologies to become more user-friendly. And we are considered that users here are potential recirculating aquaculture systems where people are actually interested on using the sources, the nutrient sources, the effluents to produce other biomass, but they are not interested or capable in becoming seaweed experts. So we hope that with the help of these kind of systems, they will not need to become seaweed experts to actually adopt the technology and start producing seaweed in this case. So with this, I'd like again to thank the partners and thank you for your attention. Thank you very much.
Thank you very much, Rui. And now is the time for Dr. Maria Roca Ayats to present her work.
Yeah, well, thank you so much for your attendance and your interest in our project. I don't know if you can see the screen properly.
We can.
Okay. Then I will follow up after Dorinda and Rui nicely introduce our production system and how we produce the algae together with the fish. I will follow up and I will explain what we are actually doing with the algae and how we are processing it to obtain highly innovative ingredients that then we formulate into seafood products and also into packaging. So basically, this aligns with the main goals of the project, of course. And I don't know if this is changing. No? Okay. So this is the overview of this part of the project. So basically, as I was mentioning, we start with the micro and macro algae that we are producing in the demo sites, together with some of the fish bite-side products such as skins and heads, and we are processing it in a cascade extraction process, as we call it, in order to obtain simultaneously different ingredients that then we use in the formulation of up to 19 different seafood products, which some of them are vegan and some of them are not. So basically, first of all, I guess that most of you already have a question in mind, it's why to process the algae and not include the algae already directly into the final seafood product. So I would say there are many answers to this question, but the most relevant ones are two. One is because of the color and taste. You may know that algae, and especially microalgae, are very strong on flavor and in color. So if you incorporate them directly into the final seafood product, then you get foods which are probably not really appealing because of the color, because they are green or brown, and also for the taste. So if we are able to process this biomass, separate the different ingredients, remove the color, remove the strong flavor, then we can incorporate these ingredients into the final product in a larger amount, so we can actually benefit from the interesting nutritional properties of algae. And then the other answer would be also in terms of a sustainable and circular point of view. If we are able to separate the different ingredients, then we can use them in a more efficient way. So take each ingredient to the final product that really requires it, and then also use the bite-side products, let's say, that we will discuss later. So now I will move step by step and explain and introduce you a little bit more on what we are doing and what we are planning to do next. So starting from microalgae, Dorinda already explained how algae can be grown together with the fish in photobioreactors. So basically, when this culture it's grown, we can harvest it in order to obtain this microalgae paste. It's actually the starting point from the processing, from the extraction process. Microalgae are unicellular organisms, so all the ingredients and compounds of interest are inside the cell. So if we want to separate them, the first thing that we need to do is to disrupt the cell and release the ingredients, and then we can move to the second part, which will be the actual separation. In here, we use many different techniques, such as centrifugations or ultrafiltrations, to be able to keep separating the different compounds. And at the very end, we are able to have 5 different ingredients that are listed here. So in one side, we have an oil which is very rich in polyunsaturated fatty acids and carotenoids. Then we have the protein fraction. We have the vitamins separately, and also a 4th ingredient that's rich in amino acids and peptides. And then we have what would be the residual fraction, which is solid, rich in polysaccharides. But instead of treating it as waste, we also use it, as you will see later, for the formulation of packaging solutions. Then for the seaweed, we also use a similar approach, let's say. And here we use 3 different seaweed, Ulva, Porphyra, and Gracilaria. And the main goal is to try to obtain protein from all of them. Also agar from Gracilaria, as this algae is very rich in this compound. And in the same way as from the microalgae, we treat the residual fraction as an actual ingredient, and we use it for the formulation of packaging, as I will explain later. Then we also treat the fish bite-sized products. In this case, we use the heads and the skins from salmons. And also we have non-compellant products. In our case, we work with surimi that it's not nice enough, let's say. But of course, all these biomasses still have a lot of nutrients, and that has to be used to be really circular. So we treat them, first of all, we green them to have this sort of paste, and then they are treated in different biotechnological processes in order to release all the protein and other nutrients. And we end up having these fish protein hydrolysates, which are very rich in amino acids and peptides. And we also incorporate them in our seafood formulations. So moving now to the actual formulation of final seafood products, we have seen how we obtain the different ingredients from microalgae, seaweed, and fish byproducts. And then we use them to formulate up to 19 different seafood products. This part of the project is just starting right now, so I will explain a little bit what we are doing and especially what we are going to do in the following months. But basically, the idea is to formulate different products, which some of them will be vegan, such as meat or fish analogs. We will also produce gummy bears, other kind of snacks like cookies or protein bars, bread spreads. And then we will have also another group, which it's what we call hybrid products. In this case, we will use both ingredients from algae, but also from the fish byproducts, so the fish protein hydrolysates. In this way, the products won't be vegan, but will be interesting and quite nutritious as well. So we will have these two sources of ingredients. And with that, as I was mentioning, we will produce 19 prototypes in the kitchen stage, and we will evaluate them. And the ones that perform the best, let's say, will be then scaled up to an industrial production. We will produce several kilograms of each one of them, because we want to perform, to test them properly and have a whole characterization of the products, not only in terms of nutritional point of view, but also in terms of shelf life, how perform compared to the similar products that are not made with algae, let's say. And also we will have a panel of experts that will evaluate them in terms of sensory performance. And also we are focusing a lot in the as Dorinda mentioned at the very beginning of the presentation, on the consumer point of view, if they like it or not, if they accept the product, on how should they be for them to accept and consume this kind of products like in a regular basis. Then finally, as I have mentioned before, we are collecting the residual solid fractions from the algae processing. And instead of treating them as waste, we are also using them to produce interesting and innovative products. In this case, these fractions, which are very rich in polysaccharides, are being characterized and mixed with plastifyers. And so we are going to produce plastic resins. And these plastic resins will be then transformed and shaped into packaging and bio-based packaging. It will be actually trays and lids. And then we will use this packaging for our seafood products. So some of the seafood products that we are producing will go inside this packaging, and then we will test the performance and the shelf life of both products. And finally, I just wanted to highlight that the main idea of this whole part of the project it's also the circularity. As I have mentioned, we are using almost the whole biomass. We are trying to have no or the minimum waste at all, and be able to take the most of the algae and demonstrate that they can be useful not only for the food market, but also in packaging and many other applications. And finally, I wanted to thank all the partners of the project. And here you have, of course, the contacts of the 3 speakers that have introduced the INNOAQUA project today.
Thank you very much, Maria, Dorinda, and Rui, for the insightful presentations. Now is the time to move for the Q&A session. So please, if you have any questions, please feel free to type them in a chat. We really encourage an interactive discussion, so do not hesitate to engage with our speaker. Now is the time. So, starting with the first question from Bert Coleman, what are the regulations regarding the use of frost water for producing food products and the use of fish byproducts for bioactive peptides and amino acids for food? Who would like to answer on this one?
I can give it a try, but the answer is very difficult in the sense that, well, actually, I don't think it's allowed. Let's say it like that. But the policies are in many ways unclear in whether they do allow it or do not allow it, because there's the wastewater directives, there's the novel food regulations. So novel food, for example, some of the algae that we use, they are already allowed as ingredients. Some of them are not allowed as ingredients, independent of whether we grow them on ROS water. Then, for example, Norway allows aquaculture waste to be used as a fertilizer, but not all countries allow that. But generally it's not seen as an animal byproduct either. So you would not necessarily have to follow those regulations. And then novel food, for example, asks again if you have a different process with significant differences to your biomass quality. So what we try to do with this project is to demonstrate that we would not have negative or any impacts on our biomass composition. But it's at the moment, I think it's the risk of the food producer, and they would have to ask then their how do you say that, their novel food, help people, so to say, whether they would be allowed to bring that product on the market or not. And I guess that's the same for the fish byproducts. The thing is very much, I think at the moment, the skins and the heads are possibly seen as waste. I'm not so sure. I don't think like the surimi, the non-compliant ones would be considered a waste already. So also there, it very much depends if something was already a waste or not. But I don't dare to say whether that could be used already. I don't know if, Maria, if you have any more knowledge here. I know we have a specific work package dealing with this, and this is actually something we're currently looking into. So what we don't know now, we hope to be sharing with
I'm not sure if
soon.
the surimi is considered waste or
No, me neither. So I would say keep following the project, and hopefully we can tell you more at a later point. Yeah.
Thank you, Dorinda. Thank you, Maria. The next question we got is from Antonis Kalaris. Great presentation. What is the production capacity from the wastewater of RAS facility?
I can start here as well. Or you, Rui?
Yeah, well, for the seaweeds, the production capacity will always be linked to the size of your production. But for the ones that we are planning, I mentioned there in my slide, so we have 200 square meters of surface area or productivity area, let's say. And for that, and based on the highest productivity species, that will be the ulva growing in there, we should be able to produce around between 500 and 600 kilos per month on average. We'll probably produce much more in the summer and less in the winter, but on average, that's our predicted productivity for that specific area, for that location with that effluent.
Thank you, Rui. Dorinda, would you like to add something on this question?
Well, as Rui says, it's very dependent on the size of the facility. So I think in Norway, your common RAS facility is for the small fish production, so to say. So there they produce around 1,000 tons of fish per year. And then if we do it very roughly, there's like 20,000 tons of nitrogen produced. So if we then look at how much algae you could produce, that's like 10 times more, but that's assuming everything can be used. That is actually also part of the project. So now we presented growing the algae on the water from the fish. We also have a task where the sludge is being treated with different processes to actually liberate more nutrients. As I mentioned also before, in this fraction of the water, we see an imbalance between NMP for what the algae need. But there's different places where we can take the water, and then we also see that those change. The ratios between NMP change, so hopefully we can use those different fractions to balance things out, and then also add those nutrients that are liberated from the sludge. So in general, I would say you can produce a lot of algae on a fish farm, and that is very nice, but that might also be a challenge in terms of how much area would you then need to if you would really want to capture all those nutrients also with your algae.
Indeed. The next one we got is from Rebecca Heaviside. Hello, Maria. You talk about using algae extracts for a range of human food products and packaging, but have you also considered the animal feed market, even recycling back to protein/lipid sources for fish feed?
Yeah, of course, this is another option. In our project, we are not considering that. Also, in our company, Algimi, we are more focused on the human market, let's say, both from food, and also we are focusing on cosmetics. But of course, this could be done as well, and this would be interesting. And I think it's something that many companies already do, but this is not our case.
Thank you, Maria. The next question we got is from Claudia Batara. Do you think GMO microalgae will be ever used to wastewater treatment and functionality ingrediated in a circular system? Integrated.
That's a very good question. I guess this very much depends, first of all, where you are in the world, because the European Union has different rules than, for example, the United States or all parts of the world. What I think potentially could be the closest version is algae being gene edited with like CRISPR-Cas technologies and those. And then there is now a discussion, I think on plants, they already have certain versions of that are not considered GMO. So I guess those, if that also starts working for microalgae and not for plants, then yes, those I foresee could be totally used in wastewater treatment and integrated in a circular system. But then we're not necessarily talking about, well, currently they're considered GMO, but at that point they would not necessarily be GMO anymore because the changes are too small and would be the same as you could achieve with traditional technologies.
Thank you, Darinda. The next question we got is from Aluvani Nemakula. Is there any current projects using microalgae formulated pellets as a fish feed instead of the ROS water?
We've had a lot of projects ongoing on using the microalgae in fish feed with very good results. In most cases, we added the whole algae, which gave good results, but similar to other plant feed ingredients. So also there we think that having a similar approach as INNOAQUA and what Maria was explaining, to have this cascade extraction and extract all the different fractions, then you could probably balance the feed much better in terms of how much protein and omega-3 fatty acids and pigments and other things you're adding. Because that was something that we saw, that if you add X% of algae to your feed, you might not add enough protein, but you might add exactly enough omega-3s, but also too much pigments, for example. So by extracting all these fractions, you can much better than define the ratio in your feed. And I guess there are still projects ongoing, but not with us at the moment.
Thank you, Darinda. The next question we got is from Ali S. Fund. Hello, everyone. Thank you for a sample presentation. I wanted to ask Maria and Rui regarding the downstream processing. if they are exploring the potential of deep eutectic solvents in membrane system for extraction of protein from microalgae. If so, which DSCs are being explored? She could provide some explanation on this domain. Thank you.
We are not doing that yet, but we actually just won a project that includes the use of deep eutectic solvents. So we are gonna start very soon. So I'm not yet ready to give any more insight because we don't have results, but hopefully in some years we will. And I don't know, Rui, in your case.
No, in the case of the seaweeds, we're not planning to use deep eutectic solvents within INNOAQUA. No.
Thank you. I have a question. With which seaweed species are you planning to work? Maybe this question can be addressed to Rui.
Yeah, the seaweed we are working with two primarily. There is the ulva or the sea lettuce, the common English name. It's the one that we expect to be more productive, and there's more information in terms of the physiology of the species. So it's a good model to be used together with the digital solution system. So ulva lactuca. So it's a green. We're going to be used also the gracilaria, red seaweed, very well known because it's now the major source of agar in the world. And as the third species, not with the same focus, but also important, we're going to be using Porphyra. Porphyra, it's let's call it a sister species of what is called now Pyropia in Asia. So the nori, the common name for the species nori that's used in the sushi, it used to be called Porphyra all over the world. Now the Asian species are called Pyropia, and our viewers in China or in Japan or in that side of the world probably know about this. So these are the three seaweed species that we are working with.
Thank you, Rui. The next one we got is from Pierre Fils. Thank you for Brian's presentation. Please, I'd like to know what the limits are of combining algae and fish production.
The limits? The limits, is that what you call them?
Yes, limits.
I don't think there are any limits. There's one thing that we also hope to prove during INNOAQUA is that the reutilization of the water coming from the seaweeds it's safe. That's part of our task is actually to analyze all the microbiome of the water coming out from the seaweeds and to make sure they are safe. And there's virtually no limit. I think the challenge here is the other way around, like Doreen was suggesting, is that typically to be able to treat or to remove all the effluents from a fish facility, typically you need a lot of area that is not available on the side of this. So typically there will be not enough area to be able to treat all the nutrients with the algae, even though they have very interesting productivities, especially the microalgae more than the seaweed. So I don't perceive any limits as long as we can prove that water is microbiological and toxicological safe. And in terms of toxicology, there's nothing there that we are adding to the water that will prevent it to be used by the fish. And in terms of the microbiome, we also don't expect that to happen because all the pathogens and all the parasites and stuff that live typically in the fish system, they will not survive attached to the seaweeds, but we'll be able to prove that during the course of the project, and hopefully you'll be able to use totally the water that is used for the seaweed production back to the fish. I hope I kind of answered the question more or less.
I hope that's true. In case you did not, Rui's email you can find on the last slide. So if you have any additional questions for Rui, Dorinda, and Maria, feel free to reach to them after the webinar. The next question is again for you, Rui. It's for Shannon Mugan. Hi, Rui. How much ulva can be produced from 1.0 hectares per year? Also, do you do any genetics improvement with seaweed?
No, right now, well, first the last part, we're not doing any genetic improvements in terms of producing GMOs, but we are selecting and screening for different strains and eventually combining different strains in one population to be able to find a good solution or the better combination of strains or cultivars, if you prefer, for that specific site. So all we are doing is just based on selection and looking for the best strain for that specific location for those conditions. So from 1.5 hectares, a round number, even though this is very specific for location, 1.5 hectares, a round number will be something between 4 and 5 tons of fresh material per month. Okay, but this will be for Ulva again, and for this kind of location and with information we have now based on the strength, a rough number will be something like this. It's a good productivity compared with other species. Microalgae, of course, have even higher productivity than this, but they have other challenges as well. So this gives you an estimate.
Thank you very much, Rui. So with this question, we came to the end of discussion and to the end of today's webinar. A huge thank you to Dr. Dorinda Klengels, Rui Pereira, and Dr. Maria Roca-Aitz for sharing their expertise with us today. It has been really fascinating to learn a bit more about the INNOAQUA project and exciting potential of algae in aquaculture and food applications. Thank you so much for joining us today. In the meantime, we received one maybe last question. I mean, it's gonna be the last question for today for Marianne. If you don't mind, I will read it before we do the final wrap up. Do you think it's possible to upscale the microalgae systems if you depend on a filtration unit like the Vibro one to separate microalgae from the water and return the algae to the bioreactor to get the new nutrients?
Yeah, but at the same time, it definitely comes at a cost. And I think it's probably not worth the cost. There is some assumptions that we made there that we might have to prove first. For example, the lifetime of those membranes. It was from a sustainability point, it was more sustainable, but from an economic point, it was more expensive. But we do think that because that was very much to deal with the how low the nutrient concentrations were. So if we manage to get higher concentrated nutrient streams, then it's actually not necessary. So we now start also working more towards getting higher concentrated nutrient streams, for example, by extracting the nutrients from the sludge. But we have a publication out on this. I can see if I can quickly share it in the chat, and then you can read all the details.
Yeah, feel free to do it. Yes, Rui.
And just a quick correction, because I was just now realizing the question was 1.5 hectares, and I gave the wrong information. I was thinking about 1,500 square meters. So it's the numbers that I gave you can multiply them by 10. That's the actual. Because I think 1.5 hectares, 10,000 or 10,000 square meters plus. So it's multiply the numbers by 10, and then you have the correct figure that I was trying to mention. Sorry about that.
Thank you, Rui. No problem. No problem. Thank you for corrections. While Dorinda is sharing the papers with us in the chat, I would like to thank you all once again and to remind you that today's webinar was recorded, and by the end of this week, I will share a link with you. And also you will find PDFs of today's slides on the EAS website. Thank you very much. I think we can see it. Yes, we can see it here. So with this, this webinar came to the end, unfortunately, but thank you very much for joining us today, and make sure you follow INNOAQUA project.
Thank you.
Thank you so much.
Thank you.
Thank you.